All right. Do we have any questions over presentation part uh, B? I'm sorry, A. My bad. A. We're gonna go over part B. I'm drinking tea, and I had mentioned something to, I think in my last class. It's recorded. You'll see it. I think that's part two, and that they're gonna keep going like that. Part one, part two. The way you've seen the others, part one, all the way down. And the final one, I'm gonna say final, or the last one or something. The word toxin, I just want to address it with you briefly, is something derived from a living entity, meaning usually toxins are derived from bacteria, or a rattlesnake venom is a toxin. And I don't mean toxic, I mean toxin. So toxin. Derived from living things. Okay, toxin is derived from living things. So I just put that in your chat. Okay, now they're living like a black widow, brown recluse, the scenario we gave of tetanus, the bacteria. But what about viruses?
Aren't they like not
They're not
living
considered
things? That's
living
when
things.
they feed off of.
Good job. Good job. So they are the non-living. Most. Are there some that are considered living? Yes, very few viruses are living, but most are non-living, because to be living, you have to be able to replicate. Now, viruses are able to do a lot of the things that living things do, but they cannot replicate by themselves. They have to use a host to hack the reproductive machinery. That's how come we call them viruses in computers, because a virus was the first hacker. Why was it the first hacker? Well, our DNA is a code. Our replicative machinery is encoded, so it goes in there and embeds its code so we can make it for it. Very clever, very clever. But it has had, viruses have had so much time on Earth before humans were ever here. So they're ahead of the game. We have only been on Earth for a very short period of time compared to viruses. So they don't make toxins, but I'm drinking the tea and it made me think of something that I had mentioned in my class. Um, I often hear things about, I'm doing a toxin detox. I'm drinking this tea, and it's a detox from detoxing from my body. Toxin. Now, I hear there are two words. Notice that I said toxin, toxic. Are there toxic things in our body? Absolutely. If we drink plenty of water, that's toxic. So the father of toxicology, which is Paracelsus Theophrastus, he said that everything can be toxic or poisonous. It is the dose that differentiates a remedy from something that can cause harm. Remedy meaning healing, something. So this is the dose, right? So things sometimes in large doses, whether they're, if I eat a lot of chocolate or if I eat anything, let's say I just decide to eat French fries, nothing but French fries for breakfast, lunch, and dinner, I'm going to tell you something bad's going to happen to me, right? Now, since we are AMP and we are talking about we talk about the human body, let's get back to this tea thing. When people say I'm getting the toxins out, they don't quite understand what they're saying, because based on this definition, they are derived from living things. Now, The human body doesn't make any toxins. None. It makes things, it can make things from cellular respiration that are called free radicals, which cause oxidative cellular stresses, like hydrogen peroxide, which is the chemical formula for that is H2O2. It's very good at disinfecting. Why? Because it turns into like free radicals that affect cell membranes, which then can kill bacteria on your wound. So we don't have toxins in our body. We have toxic things. Our poop. Our poop is very toxic. If you don't believe it, try eating it next time and see how well that goes for you. It won't work out too well for you. So nobody eats it, right? We always say the frame, it tastes like, but no, very few people have tasted that to actually say that accurately. I have, by the way, I have tasted it because I was asleep once. I think I told my A&P class, I was asleep in my apartment. Long story short, I was dreaming that I was under a waterfall and I was in euphoria and I was dreaming, I was asleep, and all of a sudden, like, there was a huge wave hit me and I woke up and I was covered in poo and pee. True story. I really was. What had happened was the person upstairs had left to another country and she was using paper towels and it all got backed up and all this poo. Apparently there was about like 1 inch to 2 inches in that apartment of poo, and it found the weak spot or the low end, which is above my bed. And I took the golden shower plus the poo. I mean, every name you can possibly think of, ladies and gentlemen. Everyone from the second floor pooped on me, peed on me. So if you've had a bad day, just think of me, okay? When you've ever had a bad day, think, I know someone who's had a pretty shitty day. And then it gets better. Man, it starts from the ceiling, just so all my stuff gets, I had nothing. Everything was destroyed. I had nowhere to live. And then you go to a college town, people, oh, I mean, there's no vacancies. I live with some senior citizens. And I lived under a dining table because they had a little dining room and it was an heirloom. And that is where I lived for for the rest of the semester underneath a dining table because it had the most peace and quiet. I could have gone to other friends, but let's just say there were quite a few people that shouldn't be living in the apartment, but there were, so they can have cheaper rent. Hard times calls for desperate measures, ladies and gentlemen. Believe me, I've had friends where I go visit them and their guys sleeping on the floor and you just walk over them. True story. That's the way it is sometimes. So yeah, that's just a side note. Again, that's a reality. It did happen to me. I have pictures to prove it. Yeah, I was covered in it. And don't think just a little stream. The ceiling came down on me, the sheetrock, everything just, and it was over. So yes. Luckily, I did not catch hepatitis. That's a very, very good thing. So I'm very happy for that. So that is toxic. That is very toxic, but we don't make toxins. We don't make toxins in our body. I just wanted to say that because there are a lot of products that claim that they do that, but they're never FDA approved. Have you noticed that? They're never the Federal Food, Drug, and Administration. They're not approved. So now, does that mean that there is no advantages to any teas? No, I'm not saying that. I'm not saying that. What I am saying that is that I don't understand the toxin removal in the human being, but there are teas that are very beneficial to you, very beneficial. Just wanted to put that thing since we are going to be looking at toxins and how they affect nervous tissue. All right, let me share that. All right, we are in B. So we're gonna kind of get into a little bit of electricity here. So like all cells, neurons have a resting membrane potential. We're gonna get more in there. If you look at that word and you see resting membrane potential, you can take a few things from here, but if you don't know what it means by looking at it, it's okay. It's all right. One thing is that resting meaning maybe not in action, potential meaning having the potential to do something and maybe taking place somewhere along this membrane. So if you can kind of get those pieces, you're on the right track. Again, what makes neurons, it says, unlike most other cells, neurons can rapidly change resting membranes potentials because they have to, because they are highly excitable, right? We were talking about Eric firing and Molly doing the controlling and sending inhibitory neurotransmitters to communicate with Eric. Yeah, it's happening very fast. So as a result, that resting membrane potential has to change fast because Molly and Eric were neurons and they are both highly excitable. They can be both highly excitable, which requires lots of ATP energy. Now, this is true. Opposites do attract. Opposites do attract. I mean, I don't care how you want to slice and dice it. It does, for better or for worse. Now, energy. When we look at opposite charges, a positive and a negative, energy is required to keep them separated. Don't believe me? Go get magnets, north and south, what we call north and south, right? And you get them and you feel that pull right when you get here and it wants to go, right? Some of you, I'm sure some of you have played with magnets at some point in your life. And magnets are so cool. Give them to kids. Of course, not little ones, like not little pieces. I mean, good sized magnets. Oh man. There's these magnets called I think they're called tile magnets. I have these, I give them to my son and it's awesome. Like he can make all sorts of things. They're like triangles in different shapes and they connect and you can make, I think they're way cooler than Legos. I really do think they're awesome. So there's this force that wants to go, right? And you have to use the energy to keep it away, right? Well, think of that. And the same principle applies to here. Energy is required to keep opposite charges separated across the membrane. Okay, cool, we got it. Well, if energy is required to keep them separated, what happens when they come together? Energy is released, liberated, right? When opposite charges are separated, it has the potential or potential energy, which is stored energy, right? Potential energy, stored energy. Water behind a dam, and the dam is not allowing water to flow, then the water behind the dam has the potential to make hydroelectricity. So when when opposite charges are separated, it has the potential to be liberated and energy released. Okay, well, definitions. Let's get into this. A voltage, a measure of what? Potential energy generated by the separation of charge, just like we talked about in this definition here. Energy required to keep opposite charges. Okay, well, that is this definition. The potential energy generated by a separation of charge. If things are separated, there must be two definite places that we can call A and B, or two definite locations. If something is separated, then they have two different points in space. As a result, well, voltage is measured since it's separation between 2 points because it's separated. So in volts or millivolts, right? Well, this is then called potential difference or potential. Now, charges can diffuse across a membrane, resulting in the potential energy. And the greater charge difference between the 2 points in space equals the higher voltage. And this all gets real important once we start firing around in the neurons. And it's very similar to electricity. It's actually the exact same definitions for electricity. Any of you guys do electrical work? Any or done any kinds of electrical work? It's the same principles. Same principles. Okay, let's go back. Current. If we're going to have these two charges separated, what's going to happen when these things are able to move between the 2 points? That's called current. The flow of electrical charge, and electrical charge is ions, positive or negative, between 2 points because they were separated. And now that they're coming together, you're going to have, now that the charges are going to be trying to attract, you now have a flow of electrical charge. And this current can be used to do work or send messages. This flow of the electric charge is dependent upon the voltage, which is the separation between the two things from the 2 points. So the flow is dependent on voltage and the resistance. Now, we're talking about flow. And when we said that the myelin sheath, the non-myelinated fibers are slower, this is why. Because myelin sheaths serve as an insulator and then secondary protector, but primary insulator. So therefore, the myelin sheath is a substance with high electrical resistance, which results in a way faster communication. Here, conductor, substance with low electrical resistance. We take a look at this again in electricity or anything like that, known as the Ohm's law. A relationship. What's a relationship? It's like an equation. So how can we put current, voltage, and resistance? How are they related, or how do they interact? Well, the current, which is the flow of ions between point A and B, is dependent upon the voltage, the potential to move between A and B, and the resistance between the path of A and B. So current is directly proportional to voltage, and the greater the voltage, the more potential difference. Therefore, the greater the current. So current over here, it would then be inversely proportional to resistance. Now, here is why I was telling you, please do your chemistry gap finders. And we're just starting now. Let's take a look at the role of membrane ion channels, meaning they are in the membrane and they're going to bring ions, meaning charged particles or elements, in or out of a cell, like we could say like a cell body, or pass into the membranes, because that has to happen. So large proteins serve as selective membrane ion channels. Now remember, proteins are made up of amino acids. Proteins, all these different proteins have the ability, and here's where the chemistry comes in. These proteins have everything in common except for one thing. That is the side chain. They have the amino group. They have that carboxyl group. They have the hydrogen. In the center is a carbon known as the alpha carbon. And above, the way it's denoted in most books, probably in your modules, it is called an R group, sometimes known as a functional group. And it is that dictates the characteristic of the amino acid. So some amino acids are water loving. Some amino acids based on that structure are going to be water hating, meaning they're going to be hydrophobic. Some amino acids are going to like lipophilicity, like liposuction, philicity meaning attraction or love, meaning love for fat, right? So as a result, proteins are the perfect thing to embed into a cell membrane because it is only really proteins that have so many different characteristics that they're able to be cool with the intercellular fluid of the cell and the extracellular fluid of the cell and be able to interact on the outside and at the same time be different in the inside, so something can then go through that protein into the cell. It's the only option most of the time because it's so unique. It's like the movies Transformers. Proteins transform to form these structures. And some diseases, most diseases, end up screwing up with an amino acid. One amino acid off, man, you could have problems. For example, cystic fibrosis, ladies and gentlemen, cystic fibrosis, that where you have that, the mucosal escalator is no longer working, so it doesn't move all that debris and mucus. And what happens with cystic fibrosis, because it gets a protein gets knocked out and the individual doesn't really cough, it doesn't come up, and all this mucus builds. And inside mucus, if you have a lot of mucal buildup, you create a bacteria, and then that leads to a lot of other problems. Sometimes they can get a lung transplant, sometimes it works, sometimes it doesn't work, but that's the amino acids. So it's because of those proteins that allow for what we're going to talk about. But I always think of proteins like Optimus Prime, Bumblebee, Megatron, you name it. Like, I'm always thinking about these things because they are able to conform, but if you change them up, it's like Bumblebee, right? If you take a critical part of Bumblebee, look, why can't Bumblebee talk, right? Let's say Bumblebee's an amino acid, right? His voice, whatever machine thing inside, it got damaged in battle, right? So when they were on the planet Cybertron. So as a result, let's say that amino acid was taken away and Bumblebee can't talk because structure is function. And if that structure is gone, he loses the function to talk. The same thing applies to science in terms of proteins. For those of you who have never watched Transformers, I am so sorry, and I deeply send my apologies because you're probably like, what is he talking about? So I do realize that, but I have to try to make bridges and connect them. So
the actual Bumblebee movie is really good.
Yeah, I like it too. I do too. Yeah, I've seen them all. I really like Transformers. My favorite one's the part 1.
Yeah,
that is my ultimate Transformer movie.
The connection you made actually makes sense. Helps to understand better.
Thanks, Eric. I appreciate that. So that's the game of proteins, and same thing with enzymes. And the reason I took that time to explain it, because oh boy, are we going to see proteins a lot. So ions, they are charged. So potassium ion channel, okay, a channel could be a protein, allows only potassium to pass through. So when you hear that, potassium ion channel, only allowing for protein. Now two main types of ion channels. One could be they call it leakage channels, which are always open. Think of a faucet that just keeps dripping. Bloop, bloop, bloop. That is a leakage channel or non-gated. Now, if you have a non-gated, you're probably going to have a gated channel, right? What does that one do? Again, here we go to the Transformers. It is the part of the protein that's capable of changing its shape to open and close. It changes its three-dimensional structure to open and close like a gate. It can let things in, it can let things out, it could stop things from letting things in and out. Now, three main gated channels, okay? A subgroup of here. Some, they call it a chemically gated. No, yeah, we can call it chemically gated, voltage gated, or mechanically. So it could be chemical gated. It really should be, I don't like it. I rather use ion gated. Ion's a better word than chemically, but nonetheless, voltage gated or mechanically gated. Okay, let's take a look at this one. I like this one way better. Ligand, here we go, ligand gated. I like it better, but only open when binding of a specific ligand, or we'll say chemical. That means that, let's say a neurotransmitter is sent from a neuron and it's traveling, well, that shape of that neuron is going to be so darn specific that when that neuron travels to the site of the other membrane, heck, let's go back to Molly and Eric. Molly was an inhibitory neuron. Molly sent a message to Eric in the form of a neurotransmitter that is very specific in its structure to communicate with Eric to cool it down and stop firing because the body starting to twitch a little bit because the toxin of Clostridium tetani is running through my CNS. So she tries sending him this, but the bacteria reaches Molly. Now, how were Molly and Eric communicating? Well, on his membrane, he had a receptor that was specific for Molly to communicate. And if Eric's antenna that's going to attach to Molly's receptor, if that is damaged, he can't do nothing. He never gets the message, right? Or if Molly is damaged, she can never then send the message. So going back to this right here, if in this gate, it is the neurotransmitter that then binds to the membrane protein, and it has to be a protein. It doesn't say it's a protein, but it has to be a protein because it is only proteins can transform. So it comes, attaches, and when it attaches, it literally changes its structure and boom, allows it to enter and then closes because it's no longer binding. Very cool stuff. Or what happens is the neurotransmitter doesn't go in the cell. The neurotransmitter comes, attaches to the side of the protein. When it attaches to it, changes its transformation to allow something else to enter. And once that something else then enters, then a signal is sent back to the membrane in a voltage, allowing the transmitter to say, oh, okay, and it leaving and then changing the protein and closing the gate. That is how these protein pumps or protein channels work without getting too crazy. That's how they work. Transformers, because they literally transform themselves in shapes. Voltage, that's another one. It could open or close. Again, that protein is going to change its structure because it receives some sort of voltage, a certain signature, and it's going to change it. And it changes the membrane potential. Mechanically now, this one's different. This one is nothing. It's not a voltage. It's not a neurotransmitter. This one responds to a physical deformation of receptors. Physical, as in a sensory receptor. So it's there, it's physical, just showing that there are differences. Okay, let's take a look at, let's say, the chemical or ligand. A ligand. What the heck is that? I was just reading what Sabrina said. Uh,
Julian,
go ahead. Go ahead.
isn't it a thing that like changes the shape of the cell or something like that?
Close. Ligand, something that binds to a receptor. Ligand. Ligand, something that binds to a receptor. Okay. Well, this neurotransmitter can be a ligand. So let's take a look. Now, I don't really like these images because they show proteins to just be these blobs, and proteins do not look like that at all. I'll show you what proteins look like. A receptor, see that little spot right there? This is a protein, and here's the phospholipid bilayer. Here's the phospholipid head, the tails over here. They have polar characteristics, non-polar characteristics. These little spheres here, the polar head, they're water loving. They have so they are polar, and here they are lipid loving, so they are non-polar. Here's a receptor, and we need this protein to open up because sodium needs to pass through here. So what do? What can happen? Well, something in the brain, maybe Molly and Eric, they have to be communicating, and Molly takes a little bit of time off so Eric can fire, and he fires off a neurotransmitter. Okay, there's Eric's neurotransmitter right here. He fires off the neurotransmitter because there is some sort of feedback sent to the central nervous system into the brain that then says, hey, I need some salt. I need some salt. That's sodium, right? Positively charged. Okay. So it cannot go in here. You may be wondering, why can't it just do that? Well, sodium plus charge is never gonna ever go through that. It needs a way. It needs a channel. So the neurotransmitter is created, and when it attaches, it changes its conformation, changes its structure, and allows for sodium to enter. And as sodium enters, potassium is exited. Now, what happens is when this stuff starts happening, the membrane begins to get charged, negative and positive, negative and positive. Now watch this. It has a membrane voltage. Now, so this one here was ligand based or chemical based. Where's that up here? Okay, there we go. Chemical based. Sorry, I wasn't showing you that. Chemical. This situation here is separate. Okay, this is now voltage. So this here's another story. This was over here, the neurotransmitter. Well, voltage gated. It's trying to go through, but look, it's blocked here. Somehow, some way, it's blocked. Okay. Well, if the voltage is changed, you see that negative? If we can get it, if we can get the negative over here, then we could change this, and this right here is going to move up. This is going to move up. Again, there are amino acids with different charges creating a negative charge, and since opposites attract, bam, that positive is going to get pulled to this negative environment. Right here is kind of neutral, and then boom, it's going to come out. That is a voltage gated ion channel. So basic principles of electricity: when a gated channels are open, ions diffuse very quickly across. And they always, ladies and gentlemen, all concentration gradients go from a higher concentration to a lower concentration. So they go from high to a low concentration. So from high to low. Now, again, along electrical gradients toward the opposite charge. So we saw that here, the opposite charge take place. So it is always easier to flow from a high to a low. For example, you go to a concert you want to go to the main stage. You want to be there at the front. But there are so many people. So many people. You got there late. You've been waiting for years to see this individual sing, play their band. Well, now you have to try. It's so condensed in there, so difficult to go all the way from the back all the way to the front at the front stage. But if there was less people, you're gonna move quicker, right? So it is always easier in nature and chemistry and physics to go from a high, something with a lot, to a low. Okay, don't believe me? Take a knife, boom, poke your tires. There's pressure in there. It's concentrated, but there's less pressure outside of the tire. So if one of you decide, which please don't do it, but if anybody poked your tire, it proves that it's going from a higher concentration of air to something that doesn't have that much concentration. No one has ever in the history of wheels ever had a flat and air goes from the atmosphere into your tire. It's never going to go from a low concentration of air to a high and fill up your tire. Never ever going to happen, right? If that example doesn't help you from highs to lows, the next time you're cold, think of me. Why? You're cold because you have energy. And energy sometimes is in the form of heat. And the temperature outside of your body is going to be cooler. So as a result, it's the thermal laws of dynamics. You're going to diffuse your heat to the open space of your atmosphere, and you're gonna lose the battle. You are never, ever going to heat your environment. You can't do it. So we have to wear a jacket or something to help insulate us. So we always go from a high to a low all the time, highs to lows. Now, when you hear things like reverse osmosis for water filtration, that's a different ball game. Now we are doing things and pushing it beyond a membrane, but that is for water purification purposes. So we're doing the opposite so we can have purified water. That's reverse osmosis, because osmosis is, again, from a high concentration to a low concentration. Osmo meaning water and going from high to low. So please remember that one. Any questions on that type of stuff? How many of you are like, oh, man, the brain, forget the brain. I don't need the brain. Darn brain, neuroscience, chapter 11, I hate you. How many of you hating it right now? It's okay, let's be honest. Teresa's hating it. She said thumbs up.
I feel like it always feels like it's a lot when you're during lectures, but when you're doing your homework, you're able to like go back and it kind of makes sense.
Okay. Please look at the videos again. I know sometimes I talk fast. Sometimes I'm going to talk really low to change my tone purposely. Sometimes I'll come to lecture and I'll try to do a deep voice for you, just change it up for you, and then go back to my squeaky voice. So yeah, I realize that it's a lot, but I promise you, like I'm doing the best I can with all my crazy little child mind examples. And I know they don't work for everyone. I know I can't, all our minds are different, but when you're freestyling it and I don't like to prepare for lectures because I just want to just freestyle, and that's the way I like to do it. So, but again, there's a lot of things out there, so many resources out there, so many different instructors you can find online, so better, they're way better than me. You can always like look up different things from different people. and you can get a lot of information. So we have a wealth of information out there, ladies and gentlemen. I know I did a lot of examples, but gosh, we really need to understand that high to low. Okay, we already did Ohm's law. How far do I want to go here? Okay, bear with me. Okay. So there are, yes, voltmeters. And what a voltmeter is going to do, we're going to measure a change. And it's really measuring the potential difference across a membrane and of a resting cell. So resting membrane potential of a resting neuron is approximately minus 70 millivolts. What does this mean? That means that your cytoplasmic side of the membrane is going to be negatively charged relative to the outside. So if you want to say cytoplasmic side, you can say intracellular, okay, negatively, and extra, like extracellular outside, all right? So you see cytoplasmic side of membrane negatively charged relative to the outside. Okay, so an actual volt differences vary from about negative 40 millivolts to about negative 90 millivolts. And as a result, the membrane is said to be polarized. Polarized. Like we call, remember we call water is polar because it has a difference in charge. So we're going to call it polarized. Potential generated by differences, right? Because we also have to have, you're gonna have the potential to do something. We're still talking about those two distances. So we can have differences in ionic consumption and difference in membrane permeability. Let's take a look at this, because pictures are way better. Let's say we get this voltmeter, and in case you're wondering, how in the world are you going to understand? Are you going to connect this to my neurons? Like how do we understand these principles? Let's see if I remember correctly. How would we do this? Let me see if I can go into the past really quick. C. elegans. Okay. I'm going to show you something. Here's a publication. These are publications in Google Scholar. And let's take a look at that. It says C. elegans longevity pathways converge to decrease mitochondrial membrane potential. We're talking about that. Ionic basis for the resting membrane potential and action potential in pharyngeal muscles of C. elegans. Interesting. Interfaces probing the membrane, a protein, blah, 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 of membrane potential, GFP molecules, sites of the neural cells of C. elegans. Action potentials contribute to neuronal signaling in C. elegans. What in the world is a C. elegans, guys? Do you know what a C. elegans is? Clearly, all these publications are doing work on C. elegans. What is a C. elegans? Wonder how we're learning all this information. There are different types of species, and they vary in size. Some of them are very large, very large. They're not like microscopic. I mean, you can pick them up in your hands. Large worms. And that is the organism in which we have discovered and learned lots of neuroscience from those C. elegans. They are the things in which we understand why. Well, look at their shape. Isn't that there's something very interesting going on there? They have very long, their nervous system is very long. It doesn't really branch out into much peripheral. They still have a central nervous system going on through that. So we can physically tap into them, specifically the larger ones, hold them down, brace them down, and probe them and study resting membrane potentials and understand how things fire and all that stuff from those worms. That is how we know a lot of our neuroscience is from C. elegans. Where I graduated from, where I got my PhD, they did a lot of research with C. elegans. Dr. Charles Martin, if we look him up here, his lab, Dr. Charles Martin's lab did a lot of C. elegans research. So we take a look at this situation. It's a voltmeter, and we're putting it inside. We're gonna, it doesn't say C. elegans, but let's just say it, you know, that's just where it's probably coming from. We're looking at these C. elegans, and we're gonna probe them, and then we're gonna ground this on the outside of some of the cells. And what that's gonna do is it's gonna allow us to understand what in the world is the difference in this plasma membrane and versus the inside. So known as a membrane potential. Let's see. Now, just in case you're wondering, the ECF, the extracellular fluid, and the intracellular fluid, I know it doesn't mention that there really well. Differences in ionic composition of just ICF and ECF. Well, we're talking about intra, and extracellular, just so you know. Let me just, and I can't change that. Let me look at it real quick. Hold on. I really don't like when they do that. I'm gonna edit that real quick. And extracellular. Intracellular fluid, extracellular fluid. Now that's recorded. Composition differences of the ionic, meaning charged, composite. There, that reads better. Differences in the ionic charge composition of intracellular fluid, ICF, and the extracellular fluid. Okay, now we can take a look at the picture. Now we can see that, oh, okay, intra and extracellular and the difference of ionic or charge. Boom, negative 70 millivolts. Now, differences in this ionic charge composition is that the extracellular fluid has a higher concentration of sodium than the intra, right? So since it's higher, well, guess what? That sodium is gonna wanna come in. That's what we looked at the other one, right? Now, it's balanced mainly by chloride ions, okay? Chloride ions. Now, the intracellular one has a higher concentration of potassium. So remember this, that the outside extracellular has more sodium, the inside more potassium, and this one's going to be balanced inside by charged proteins, charged amino acids, right, within the membrane. This one here, since that's a positive, it was going to want to be balanced with chlorine. Now, potassium plays the most important role in the membrane potential. Potassium plays the most important role in the membrane potential. Now, if you notice that this is a positive, maybe difficult to see, it says balanced mainly by sodium chlorides. As we move on later, you're going to understand that there is a sodium potassium pump, and then you're going to see that there's always this balance, this affinity between charged particles. In this case, the same thing applies negatively charged proteins, and where the protein is going to be embedded. There'll be peripheral proteins within the membrane. A principle of physiology is that the body is always going to try to find balance. So if something is a negative charge 2, it wants to give a positive charge 2. Sometimes that is not a good thing. Sometimes there are certain diseases and certain things or certain chemicals or certain narcotics that are charged and cause certain things to happen. Some situations, an individual has brittle bones at some point in their life. Why? Well, inside bones, you have calcium. Calcium is charged, and there is something that is releasing from the bones to find balance with the charged particle. Please remember that principle. The opposites attract. And sometimes if there isn't this balance between the charged particles, positive and negative, sometimes bad things happen. So in the body, we're always trying to find a balance. Homeostasis, right? Homeostasis. So I'm gonna leave it with that right here, where we went through all this. Let's take a look. We went over resting membrane potentials. We talked about neuron excitability. I gave you the principles of electricity. We go voltage, potential differences, currents, resistance, Ohm laws. We talked about transformers, the leaked ones, always open, the gated. We talked about chemically, also known as ligated. We talked about the voltage. Now we went into voltage. Now we're understanding a little bit about voltage, but that still involved a protein. Then the mechanically gated ones. We gave you examples here with a neurotransmitter one in which it attached. We gave you the voltage one. Then here's a mechanical gated one. So examples of the gated and the voltage. I talked and gave you examples of things moving from higher to lower concentrations all the time. Gave you Ohm's law. We talked about this -70. I came here, did some editing with intracellular fluid and extracellular fluid, gave you an example of C. elegans, in which this is how we truly learn stuff. Now, have we put voltmeters in people's brains? Yes, yes, we do. Yes, correct. But before we do that, we want to get some information first on C. elegans. Here, talked a little bit about, hey, what are we finding on the outside? High concentrations of sodium. What is that being balanced with? It's going to be balanced with some chloride ions, opposite charges, right? Okay, in the case of the inside intracellular potassium, what is that being charged with? Charged protons. I'm sorry, charged proteins. And those proteins are made up of amino acids, and some of those amino acids are going to be negatively charged, so they can help balance that. Potassium plays a major role. So I'm going to leave it with that. Please take time to look at the videos. The videos I'm going to post up tonight. Replay it as much as you can. That way, this part gets hard. This part gets a little hard, and then it gets a little better. And then it's going to get even more better. Like, we're going to get into blood, and we're going to get into the immune system. We're going to go into the lymphatic system, the digestive system. But you have to get this piece, because chapter 12 is central nervous systems. So you got to get all these foundations to make sure you have your 11 and 12 knocked out correctly. So this is hard. Not gonna sugarcoat it. It is. I go very in depth here, but I have to go in depth here because this is kind of a little hard to learn by yourself. So I'm using this as a guidance. There are going to be other topics that you're going to be like, oh boy, I got it. If I can see that on a PowerPoint, okay, I get it. Like, there are going to be times like that. But then there are going to be times like this here. This is neuroscience, and we have to talk about this one because it's this peripheral, the central CNS and the PNS that's going to do a lot of controlling for the rest of the organs. So we're going to start going into other things. But if you feel like, whoa, that's okay. If this is the first time you're looking at PowerPoints, you're going to have the whoa factor. If this is the first time you're looking at any neuroscience videos that I posted, you're going to have the whoa factor. That's why it's real important, ladies and gentlemen. Please try to just picture walk through the book, through your e-text, anything, scroll, anything to help you come to this theater and kind of sit back and be like, okay, I don't understand everything, but your brain's picking up more information because it's your second dose, your third dose of exposure of images. When I try to explain something and it's the first time you see the image, it's like, for example, if I get something like if I get this is for my tea, and I go like, well, whoa, you may not, first, second time, but if I go, hey, whoa, it's honey. Okay, you're kind of getting it. You saw some color, you saw the image, and now you're saying, oh, that's honey. Because the third, fourth time of seeing it, you kind of get a better idea. So that's to remove any kind of like, oh man, you don't have to read the whole chapter. I do a lot of the legwork for you, a lot of it, but try to look at the pictures, definitely. Even if you don't understand the picture, just look at it and look at the picture and say, oh, what are the parts of the picture? You don't have to know what the parts do. I'll explain that for you. That's my job. But try to come to class knowing a little bit about the picture. What is it made? What is it made up of? What are the parts? And then I'll tell you what they do. Okay. And then leave that part up to me. All right, ladies and gentlemen, do we have any questions?
No, sir.
No, sir.
All right. Well, everybody have a wonderful evening. Take care. And I will see you all on Tuesday. All right. Bye.
Bye.
See ya.
Bye.
Bye-bye.
Thank you. Bye.
Bye.
Bye, sir. Have a good
See ya.
day.
You too. Take care.
Bye.
Bye-bye.
Good night.
Good night. See you, Omar. Take care.
Mr. Delgado.
Yes.
Oh, I got the COVID vaccine yesterday.
Oh, that's good.
Yeah.
How are you feeling?
I had no side effects, just my arm's still sore. That's it.
Yeah, my parents had it. My parents got it on Monday, this Monday, and so far they're good. Nothing happened. Do I know? I know some people in Incarnate work at the medical center there, and I know some of the staff there. And
Yeah.
some people had little symptoms here and there, you know. On-
Yeah,
Yeah.
I just got the second dose on Monday.